So it is my pleasure to introduce Dr. Valafar. He is a professor of Epi and Biostats at SDSU, and before joining the School of Public Health at SDSU, he was a professor of computer science until about 2019. And before SDSU, he was an assistant professor of cognitive and neural systems at Boston University. There he was from 1999 to 2001. He's a NIAID PI leading projects on global pathogenesis and diagnosis of antibiotic resistance of TB. This project involves diagnostics, prognostics, evolutionary analysis, molecular epi systems biology of TB, and drug-resistant TB all together. And this is what I think we're going to learn a lot about today in terms of how TB becomes resistant quickly. And it requires all those things to really understand how this works. The clinical component of the project includes diagnosis, sample collection, and treatment of TB in 8 high TB burden countries from Europe, Asia, Africa. And he has microbiologic components of the project in connection with the WHO and their reference laboratories in Stockholm and Antwerp. And of course, he does lots of, based on his computational backgrounds on silico components that he conducts out of his lab in San Diego. And Dr. Benson will be moderating the QA, one of our local the experts on the UC, Dr. Valafar's presentation. Okay, and then I'm going to turn it over to Faramarz. Thank you.
All right, thank you, Davey. I much appreciate the introduction. I want to thank you for the opportunity to talk about our research here. Davey gave a really comprehensive overview of our project, so make my first 4 or 5 slides useless, so I'm going to speed through those. All right, so as Davey mentioned, we are an NIAID funded group. We do international work in understanding evolution of tuberculosis, emergence of drug resistance, and other clinically relevant phenotypes such as persistence. We have some clinical sites from which we collect clinical specimens, and we have some microbiological sites where we do all of our BSL-3 work. What is new is that we have a BSL-2 here in-house at San Diego State to do some work relevant to TB, but clearly not on the organism itself. So a bit about HIV and TB. TB is, I still call it the most deadly infectious disease. This is still, I believe, to be true in the era of COVID-19, and this is simply because we have over 20 vaccines, most of them very effective for COVID-19. For TB, we don't have a single one, at least not an effective one. TB and HIV, they combine to a perfect storm. HIV positive patients are 26 times more likely to develop active TB. Of the 10.4 million people who develop active TB each year, 1.2 million have also HIV. Now, we thought we had a good understanding of TB, at least from its prevalence, but a recent report from 2018 showed that we really don't have a good understanding, and it demonstrated that the prevalence of TB is quite a bit higher than what we originally assumed. And one of the factors that I just wanted to point out with this slide is I wanted to raise awareness to subclinical TB, which resembles COVID asymptomatic cases. A paper from 2020 was published on this subject, and as you can see, there are some cases, they have this nice figure. I'm not sure if you can see my cursor. They have this nice figure where they show latent TB, but they also show a novel phenomenon called cyclic disease, where in some periods it is clinical, meaning that the patient demonstrates symptoms, but in many instances, it is subclinical. There are no symptoms. However, the patient remains infectious. This is something interesting that we just haven't really studied all that much. So it sounds like COVID. What is important, though, for this audience is its interaction with HIV. Importantly, emergence of drug resistance is a major problem globally, but that emergence, the pace at which drug resistance evolves is higher in HIV-positive patients. And unfortunately, the state of diagnostics, even though we do have rapid molecular diagnostics, is still a whole lot of places around the world. They use culture-based methods to diagnose drug-resistant tuberculosis, and outbreaks in South Africa have been documented to have killed patients while on standard therapy, waiting for culture results that eventually indicated drug resistance. A very good article came out in Lancet Infectious Diseases about the transmission of drug-resistant TB, about the role that that rapid molecular diagnostics could play, how the chemotherapy shortens. But what this ultimately showed, this article, is the interaction, is the transmission of TB between HIV-positive patients and the general population, evolution that happens throughout that transmission, and then back to, from general population to the HIV community. So to really understand all of this, we really need to develop better diagnostic methods, and that automatically puts us in the TB genomics domain. And we do need to understand how drug resistance emerges. We need to understand its mechanism or mechanisms, as well as the speed at which these mechanisms emerge. To do that, I wanted to cover some basics of TB genome. The Mycobacterium tuberculosis genome is an unbalanced circular genome. What that means is that it has higher content of a higher GC content, 62.5% in fact. And also importantly, unfortunately, we don't know a whole lot about this genome. We know the function of many genes, but that functional assignment is often partial, meaning that we don't know everything that that gene does, what pathways it involves in. And about 40% of the genome is also genetic dark matter. We have no clue what that does, what that 40% does at all. And so when we do molecular assessment and when we do whole genome association with the phenotype, we often end up with mutations in regions that we don't understand what they do. So I also, before I get to the next slide, I wanted to point out that in genomic communities, Mycobacterium tuberculosis genome is considered boring, mostly because it is assumed that there is very low rate of mutation. Most of the genome is stable, so there is not much to study. However, clinical reports indicates otherwise. A very good example of this is we collaborate with Belarus, a country that is doing a whole lot of clinical trials for the World Health Organization because they have a lot of MDR TB there. And a few years ago, when bedaquiline was being introduced, a couple months after that introduction, we got a call that there are some cases that don't respond to bedaquiline. And at the time, phenotyping of bedaquiline was problematic. Midget, which is a standard phenotyping platform, wasn't doing well. The drug was binding to the sides of the chamber. And so there wasn't really a good way of determining drug resistance. And so what we now know were that all those cases were resistant to bedaquiline. The main question is, how did resistance emerge that quickly in a pathogen that is one of the slowest growing pathogens among bacteria. And so there are a lot of unanswered questions after so many years of us as a species battling with tuberculosis. Now, here's what we do know. We know the genes that are involved in resistance to some of the major antibiotics that we use. We also know which mutations specifically in those genes cause resistance, or at least are associated with resistance. And we also have a fairly good understanding of, if we use those mutations in a molecular assay, what percentage of resistance do we catch? And the percentages that you see indicate that those are the sensitivities. Now, there's a whole lot of activity going on all over the world to discover new mechanisms that would provide new mutations for these molecular diagnostic platforms in an attempt to increase these percentages. And my lab is one of those that constantly we are looking to identify these novel mutations. Now, what I want to discuss is some of the challenges first. First of all, we published a paper on Illumina platform sequencing Mycobacterium tuberculosis genome. We chose Illumina because it's the most prevalent platform that sequences MTB genome. And the reason we did that is because it is well known that short read sequencing that uses amplification before it's the actual sequencing doesn't do amplification properly. And so some parts of the genome are amplified uniformly. And so our systematic study of this phenomenon resulted in some very interesting findings. For the first time, systematically pointing out which parts of the genome aren't amplified and therefore aren't sequenced properly by Illumina. The red circle that you see indicates where in the circular genome there are issues and Illumina platforms cannot sequence those regions properly. And the other colored circles, for example, the light blue shows where repeats are in this genome, or where the high GC content is, that's the dark blue. And you can see that many of the blind spots coincide with those. And we already knew before this exercise that repeats and high GC content causes blind spots. At least now we do know which parts of the genome suffer from this. The other thing I wanted to point out about this plot is that there are also some blind spots that are not explained either by repeats or GC content, meaning that other genomic sequencing attributes also contribute to blind spots, which we don't quite fully understand yet. And here is the breakdown of which genes are affected by these blind spots. As expected, a whole lot of PE and PPE family of genes are affected. That's the dark blue. That was not new. In many studies, people in fear of these blind spots, they just throw those parts of the genome away, which is unfortunate because those genes have a whole lot of very interesting and clinically relevant function, and we do need to study them. But also what we were able to show is that the hypothetical genes, that's the dark region of the genome, which we don't know anything about, also quite a bit suffers from these blind spots. And if we want to study and shed light onto those parts of the genome, we really cannot rely on Illumina sequencing to do that. Here's a list of genes. This is sorry, do you guys hear the repeat?
I don't hear a repeat.
Oh, I can hear my own voice repeating.
Oh, I don't know. Does anybody else hear it?
Yeah, no, I guess I'll just move on. Okay. Sorry about that. So the previous slide had some genes listed. That's a subset. It wasn't a full list of genes that are involved in drug resistance, but also are suffering from blind spots. Now, the challenges that lie ahead in terms of sequencing, we really need to know which parts of the genome we are missing. And I explained a little bit about that because of the blind spots. The other question is, what is the genomic structure? This is the part that very few researchers pay attention to. And it has to do with, for example, gene order, which genes are in an operon and which are not. Previous study by my group, as well as others, have shown that operons change dynamically in response to the hostile environment that these bacteria run into in lesions and in other parts of the body. In terms of acquisition, we cannot explain how resistance is acquired so rapidly. And I'm hoping that to show you some slides about the mechanism that we've discovered that I think has the potential of explaining the rapid emergence of drug resistance. Clinically, first, which parts or where in the genome do we assign a phenotypic role? What changes in those regions cause a phenotypic change? In other words, if I identify a gene, for example, gyrase, that plays a role in mechanism of action of fluoroquinolones, which mutations in gyrase cause resistance to fluoroquinolones? The answer to this question we mostly know, but similar questions for novel mechanisms persist, especially the new and repurposed drugs that have come recently into the market. Some changes are associated with, but may not cause phenotypic change. For example, we know of compensatory mutations for rifampicin resistance. We can use those for diagnostic purposes, but we cannot use those for mechanistic analysis. So we need to be able to distinguish these, and simple association does not do that. And when we look at the mechanism, we run into the significant problem of 40% of the genome being unannotated. When these mutations fall in the hypothetical gene, several questions persist. Is that gene expressed? If it's not expressed, then that mutation is irrelevant. If it is expressed, what does it do? All of these questions are unknown. And all the different areas of tuberculosis research that David mentioned at the beginning, we have tried to get into those out of necessity to answer all of these questions. Now, a bit about short read versus long read sequencing. As you can see in the lower corner, I'm showing short reads as they have aligned. And you can see the red arrow showing a part of the genome that only has one read covering it, which is insufficient to confidently call what is in that region. Now, that problem is mitigated mostly by long read sequencing, which is the form of sequencing that we use. We've gotten into that using primarily PacBio sequencing, but also some Oxford nanopore sequencing. Now, when we do long read sequencing, we run into interesting possibilities, but also interesting new challenges. Now, with long read sequencing, we no longer have to do reference-based assembly, which has the problem of assuming that every clinical isolate out there has the same genomic structure as the reference genome. When we try to do de novo assembly, which is the assembly method without assuming a genomic structure, we were able to do this with a pipeline that I'm showing without getting into the details of it, but we also were able to show that the genomic structure significantly varies from what the reference genome has. And so some of the examples of that I will show in a minute. This is a bit more about the de novo assembly pipeline. I just want to mention that what this pipeline does, it takes raw reads that come from out of the sequencer, it circularizes them, polishes them, cleans up all the errors that we catch in terms of short errors as well as the structural errors. But this pipeline also provides DNA methylation, which I will get into in a little bit. And finally, we assemble the genome and are able to tell the variations that happen, the short variations, the classical SNPs, as well as short insertions and deletions, but also structural variations, things such as inversions, translocation, and things like that. And when we let that pipeline loose on a typical exercise that has been done before, basically comparing H37RV, which is the common virulent reference genome used or reference strain used to study tuberculosis versus H37RA, its attenuated version that is not virulent. The genomic comparison of the two had been done before, and some mutations have been identified. All of them SNPs had been identified by a lab in Hopkins that they then argued that those mutations are the basis for the virulence in Mycobacterium tuberculosis. When we did this exercise, we were able to show in an article that most of the SNPs that they reported were actually sequencing errors. In addition to that, we were able to show that significant differences exist between the two strains, but on the structural front, not necessarily on short variation front. And so PacBio loved that exercise, and so they advertised it as one of their platform's accomplishment. So we've got some free publicity on that. The other thing that I wanted to mention about the structural variation is the role of an insertion element. There are several insertion elements in MTB genome. The most common one that people have studied is IS61110. First of all, there are a number of copies in every genome. That copy varies from genome to genome, but also varies notably from lineage to lineage. Now, these genomes are, they move around quite a lot. And in that process, they do all sorts of things to the genome. Through homologous recombination, what happens is that the region, the segment of the genome that is stuck between two of these elements sometimes gets inverted, sometimes it gets translocated to a different part of the genome, sometimes it gets deleted, and as a result, it causes all sorts of disruption to certain genes, and it introduces also sometimes new and novel genes. First, on the right lower corner, I want to show you the number of IS61110 elements that we see in these genomes, and this is the statistics of about 100 genomes, and the different colors indicate different lineages. And as you can see, there are some patterns, and some lineages do show greater variability, at least on this front, because of IS61110 as compared to others. The lower left figure shows where in each of the genomes we were able to detect a copy of IS61110. The blue indicates that that copy was on the plus strand, and the red indicates that that copy was on the negative strand. And these are, we did this comparison between some strains from Sweden as compared to some from South Africa. And as you can see, there are significant differences on that front. What that sums up to is that tens of thousands of base pairs in the genome are changed either in location or in direction, or even more significantly in terms of deletion, they are deleted from the genome, or new ones inserted, recombined with some other genetic material. And so on a typical genome, when you look at how many SNPs there are or substitutions, there's about 1,000 to 2,000. But these structural variations caused by IS61110, they cause tens of thousands of changes in the genome in terms of number of base pairs. And so their potential functional impact is quite a bit higher than a single SNP. Now, what we have been able to demonstrate is that one of the common functions that we see, and this is quite frequently observed, is when a part of the genome is in need of a mutation. And because of, for example, antibiotic resistance or some phenotypic requirements such as persistence in deletion, that part of the genome is often inverted. And when the transcription machinery comes in and has to run in the opposite direction, that increases the rate of mutation, mistakes that happen in this transcription process. And that is how we believe higher rates of mutation happen in the genome where it needs to happen. Now, the other point that I wanted to make, PE and PPE family of genes are highly interesting because they interact with the host immune system. And we thought that we had some understanding of the whole arsenal of PE and PPE genes. But what the slide shows, this is a phylogenetic tree of everything that we saw in the genomes. The different colors indicate different sublineages, but the gray is the more interesting genes. Those are genes that we have discovered. These are novel. They line up very closely with known PE and PPE genes, and we have made the argument that these are PE, PPE genes. It's just that they are novel. And through combination as a result of movement of IS6110, these new novel genes are created from the combination of two existing ones. Now, the challenges in TB diagnostics cost is certainly a big problem because TB is usually prevalent in very poor countries. And so the cost cannot be very high. And as a result, the diagnostic platforms that we try to develop have to be low cost. Bacteria are slow growing, therefore culture-based methodology is really too slow and cannot work. And also bacteria, importantly, what we have been able to show is bacteria that do not grow in vitro do matter. There are a whole lot of bacteria that grow and cause the disease in vivo, but they just do not have the ability to grow in culture. And so we are not able to observe those. We think most of this is because the antibiotic treatment that we provide, they eliminate a whole lot of these bacteria from their cell wall. And that pushes them into persistent in a state where they just don't grow. And unless the specific nutrients from the host are provided, they will not grow. As a result, we won't be able to see them in culture. Now, remember this slide that I showed you, the sensitivity of various mutations in genes that we do have currently. These numbers constantly go up and down depending on the type of research that people do. Now, the question really that I would like to ask is, are we looking in the right place? Structural variations are important. We have been able to show that they make significant changes in the genome. Gene order is important. Operon structures are important. They are dynamic and respond to the environment. Inversions cause higher rates of mutation and do need to be scrutinized far greater than they have right now. There's also gene duplication and deletion. A lot of people study gene promoters because they want to understand expression levels of genes. But another mechanism of increasing the expression level of a gene is to simply duplicate it. And we have demonstrated that many genes are duplicated in this organism. Now, the other thing that people usually don't look at is in the epigenetics domain. Base modification and methylation, specifically, we have looked at, and we have discovered some fascinating stuff. In terms of genomic structural variations, this slide is just to give you some sense of what types of structural variations we see and what type of variations or how frequently we see them. And you can see the different kinds: duplication, deletion, inversion, insertions, jumps, and breaks. And you can see that the numbers, they do change in terms of which genome you look at. But also the size of these variations do change. And we still need to understand what that size and the location of these structural variations indicate. We do see consistency in terms of location. So that's a very interesting phenomenon. Here is a bit more detailed statistics in terms of the size of the structural variations. We do see the mean and the range that they do cause in terms of novel genes that we do see. On average, now this is really important, we have identified 167 novel genes. These are genes that are not in the reference genome. We don't understand what they do, but we have identified mechanisms that do create these new genes. Usually they are gene conversion in terms of two genes combine to create a new gene. That one new gene does the function of both previously existing genes. And what advantage this presents, we do not know. We have some hypotheses, but it is a mechanism that does need to be studied. I can also show you that some copy numbers, these are duplicated genes. They do vary between lineages, and East Asian, that's the orange plot in the bottom, is perhaps the most virulent lineage out there. And you can see that there's a higher incidence of copy numbers. And so genes are duplicated more frequently in East Asian, especially the Beijing sublineage. Now, I hope I have a little bit more time to describe a very interesting phenomenon that I believe explains the rapid emergence of resistance, and that is on the epigenetic domain, specifically DNA methylation. The phenomenon that we have discovered and we report, we've named it intercellular mosaic methylation, and it presents the potential to create hundreds of new phenotypes every cycle, and every cycle of this pathogen is about 24 hours. Now, we published this paper on eLife. We described first the diversity of methylation patterns in 100 and some isolates that we have studied. We also identified loci in these genomes that change in terms of methylation. I should also say that in bacteria, DNA methylation motifs exist, and they are typically under normal circumstance methylated. When they do go unmethylated, that describes an anomalous condition that creates changes in phenotype, and I'll describe that in a bit. So first of all, some DNA methyltransferases, or MTAs to be short, function, sorry, let me just move some stuff around, function as the self-protective component of restriction modification system. Now, this part is really interesting to me. Other conserved, we call them orphan MTAses, have evolved alternative functions, sometimes through interaction with transcription factors, cell cycle control, for example, virulence modification, and so on. By orphan, we mean they lack a partner restriction enzyme. Now, the other important factor is that DNA methylation can mediate formation of phenotypic variants of genetically identical cells. What that means is that when you sequence and you only look at the chromosome without the epigenome, you see that two bugs are similar to each other, but where the phenotypic differences that are observed are actually driven by DNA methylation, not changes in the chromosome. Now, the interesting part here is that MTB, and I'm here talking about the species, has 3 methyltransferases. Unfortunately, H37RV, the reference genome, and it's the one that is the most commonly used, only has one of them active. And so for all DNA methylation studies, H37RV proves to be a poor model. Now, the other thing that we have clarified in our manuscript is that the third MTase, HSDM, connotes a restriction modification system, but there is no such restriction modification enzyme. And so when we sent in our manuscript, we really didn't have anything about that, but the reviewers recommended that because of that fact, we should recommend a new name for HSDM, which in our manuscript we have offered the new name of MAM-C, consistent with MAM-A and MAM-B, the other two MTases. Now, in terms of this slide, I just want to very quickly go over in terms of what our pipeline does in terms of methylation analysis. We first do a methylomic survey across many different isolates, and then we do MTase genotyping. These are the mutations in the genes that code for the 3 MTases. We do that, and then we do study heterogeneity, and we then correlate anomalous methylations of DNA in genome in gene promoters with sigma factor binding sites to understand functional consequences of those anomalous DNA methylation events. First thing that we were able to clarify in our manuscript is that methylation in MTB is not lineage specific. It actually displays convergent evolution properties, much like the mutations that confer drug resistance. This is in this clarification came through our manuscript because previously, famously, a paper by from Albert Einstein College of Medicine described these events as lineage specific. And they concluded that they do not have clinical consequences, much like mutations that are in the chromosome, but are lineage specific. We demonstrate that this is not correct, and we linked the incorrect conclusions to the fact that they used H37RV as the reference genome versus what we have done, which is de novo assembly and true understanding of where each gene is and where its promoter lies. Now, mutations drive diversity in Mtbc, and I want to show this through for each of the 3 MTases. As you can see in the slide, the interesting part that I want to highlight, let me actually move forward to, you see that arrow that is pointing to one instance of an isolate where the IDP ratios don't really line up with the active phase or the deactive or the inactive phase, and they are somewhere in between. For that arrow is showing it for HSDM or MAM-C, but there's also 3 isolates that demonstrate that behavior for MAM-A as well. Those are the light orange colored isolates. And a similar behavior is also present for MAM-B. So all 3 MTases have these isolates that don't really represent a clean picture. If the methyltransferase is active, they really need to be lining up with the with the IDP ratios at the top where the blue line is. If it is inactive, they should be lining up with the orange. We didn't understand why they didn't line up with either, and they were somewhere in between, which led us to the investigation that led into intercellular mosaic methylation. Now here's the picture focusing on MAM-A, and we were able to correlate that behavior with the genotype. In other words, the specific mutations that happen in the gene that codes for MAM-A. And so what we discovered was that quantified fraction of methylated motif sites in single DNA molecules represent this behavior of this mixed behavior. And so unimodal distribution consistent with the intercellular stochastic methylation, this is a phenomenon that was known before, this is nothing that we have done, collectively give rise to intercellular mosaic methylation. And distinct from established forms of DNA methylation heterogeneity, we show that, let me see if the next slide, yes, the next slide shows this much better. So the top 3 forms have been described before. The bottom form is described by our manuscript. And so methylation patterns do give rise to specific phenotypes. For example, the top one shows that they do persist under drug stress. And when methylation, when methyltransferases are active, then the bugs have a higher ability to survive in low oxygen levels. Phase variable methylation is something that has been described well previously, is able to provide 2 phases or 2 subpopulations that one is able to survive under drug stress and the other one is able to survive under low oxygen conditions. What we describe in intercellular mosaic methylation is the creation, is the mechanism to create hundreds of new phenotypes. Some of them are able to survive under drug stress, some of them are able to survive under low oxygen conditions, and some are able to survive under all of those stress conditions combined. And this is what we believe allows TB to survive throughout chemotherapy and re-emerge in many of the patients that suffer re-emergence of treatment. I'm going to quickly go through some outstanding questions for intercellular mosaic methylation. One is that, is there a mechanism for heritable methylation patterns in IMM? The other one is, to what extent does IMM-driven methylation diversity generate phenotypic diversity? And finally, does IMM-driven phenotypic plasticity affect adaptive evolution in strains exhibiting constitutive IMM? These are difficult questions to answer that we feel obligated to answer. Orphan methyltransferases harbor site hypervariable across clinical isolates. So we have identified which sites specifically are hypervariable across different strains. And we can correlate that then to phenotypes, clinically relevant phenotypes, much like people do with chromosomal mutations. We have discovered evidence of transcription factor occlusion at hypomethylated MTACE sites. These are hypomethylated sites are the sites that are consistently across many isolates have none or low levels of methylation. Promoter methylation is also a significant phenomenon, is widespread and consistent with CIS regulatory role in Mtbc. And so integrated promoters determined by 2 transcription site start sites, we see that these methylation sites actually coincide with these TSSs. And as a result, we believe that these cause gene regulatory functions or controls. These act like switches. We identified 33 promoters that do work in this fashion in the genome. Now, widespread promoter methylation. This is a sort of a heat map of all the methylated promoters that we show. And you can see that there is a great deal of variability, but there is also consistency across the different isolates. And so that consistency, we believe that is not a random phenomenon, and there is a advantage that these patterns provide. 212 genes with promoter MTase motif sites. Each one appeared in greater than 75 isolates. So these are, we don't believe the statistical probability of this happening in 75 randomly is extremely low. Hypervariable sites enriched in the region spanning the -10 and -35 promoter elements. These are TSS binding sites, and so those are really interesting for us to study. And promoter methylation is widespread and consistent with CIS regulatory. Again, HSDM or MAM-C methylated promoters indirectly influence transcription. We were able to show that in our manuscript. This was another point that we were able to clarify before the understanding was that it doesn't. Direct influence of MAM-C methylation on promoter strength is also demonstrated through our manuscript. And finally, promoters with MAM-C motifs enriched in the deletion of certain genes. And this is a really interesting path to go forward, making the argument that DNA methylation plays a role in structural variation of the genome. I'm going to skip this slide in the interest of time. If there's interest, I certainly would love to come back. But in conclusion, Mycobacterium tuberculosis has acquired mechanism of genomic scale change without horizontal gene transfer. This is really important because it has been shown that gene transfer doesn't happen, horizontal gene transfer doesn't happen in M. tuberculosis, mostly because of the thick cell wall that it has. We demonstrate mechanisms that internally recombine genes to do what horizontal gene transfer eventually does. There is far greater variability in M. tuberculosis genome than commonly known. Phenotypic role of such variations are largely unknown, and a whole lot of studies need to be done. And Mycobacterium tuberculosis has the ability to adapt rapidly, quite possibly within 24 hours through epigenetic arsenal. And we believe that at least those epigenetic mechanisms provide the ability to persist throughout chemotherapy or immune system pressure. And when those pressures subside, either these methylation patterns go away or in the process, chromosomal mutations emerge. We have shown that also phenotypic consequences of some of the structural adaptation and some epigenetic adaptation mechanisms are significant, and they affect some genes that involve antibiotic resistance. And finally, the question of whether such mechanisms are more active in vitro remains unanswered. So basically, the really interesting question is how much of what we just described is happening in vivo versus in vitro. And we believe that we've only scratched the surface on that front. Thank you very much, and I'm sorry I went a little bit over.
Thank you, Dr. Valafar, for that very elegant discussion. I think you've wowed us with a lot of really spectacular genetic manipulations, I guess. So I'm going to open it up for questions. And if any of the attendees or panelists have questions, please, you can either ask to raise your hand or put them in the Q&A or chat. I'm going to start off with a couple of questions that occurred to me as you were talking. And, I think your conclusions are spot on, because I think for some of us who work at the more clinically oriented interface with a lot of the discussion that you've provided with us today, it's sometimes a little bit difficult to see how this plays out in a clinical setting. And one of the points that you made, which I think is a very important one, is the variability in the genome. And we've been used to understanding about tuberculosis for many, many decades now, that the replication of the MTB genome is error prone. And so I guess my first question for you is, how much of the variation that you see in your in vitro studies do you think occurs as natural error-prone replicative consequence, or how much do you think is actually driven by some of the external influences, such as drug selection pressure or human immune response pressures?
Right. Thank you. That's an excellent question, and really important question that we do need to answer. What it looks like to me at this point is that there is a combination of both factors that you mentioned. The error in the transcription and translation machinery. But what is really interesting is that error the bug seems to have to be using it, sort of guiding it to target specific parts of the genome as a response to drug pressure and the immune pressure. Mm-hmm. One of the more interesting mechanisms is the IS6110 element causing inversions. When inversions happen, then of course the transcription machinery is going in the opposite direction, and that rate of error, which is natural to MTB, increases. Now, what is interesting is that typically causes random mutations. What is interesting is that inversion we have discovered is that it's strategically happening in specific parts of the genome. And as a result, it looks like the machinery, the organism, is actually guiding the machinery to commit more errors in specific parts of the genome. And those parts, errors in those parts present a benefit in presence of antibiotics or immune pressure. So to me, it seems like there's a combination of both of them. The natural error in the mechanism driven by the external pressures.
Great. Yeah. That's very clear. Thank you. One question to you from the audience. Are you sequencing and analyzing structural adaptations?
Yes, that's exactly what we are doing. Let me start by saying that our sequencing process is very similar to what other people's do, with the distinction that we use long reads. But what we do post-processing is unique in that we use the long reads to assemble the genome de novo, which then exposes all the structural variations in the genome. And then when we compare the genomes to each other in comparative genomics, we do short variations as well as a structural variation analysis. So that's how we have been able to discover all of what I reported today.
And then a follow-on to that question, are you able to use clinical isolates over time to analyze the structural adaptation with epigenetic mechanisms over the time course of treatment, for example, in a single individual or a group of individuals? Are you able to do that?
Yeah, so excellent question again. I'm really impressed. You guys are asking questions about everything that we tried to do. Yes, so we did get funding from NIH to do that. Unfortunately, it ran into the COVID era where everything shut down. So we do have a promise from our clinical sites to sequence bacteria from each case in a serial fashion, in a time course fashion. And that really would answer a whole lot of these questions. How mutations change in the chromosome, how DNA methylation changes from one part of the infection to another, but also the order in which these changes occur. Our hypothesis is that DNA methylation changes occur, and where they occur, they increase the possibility of chromosomal changes in those loci. And so whether that is true or not, we are hoping to be able to answer through these time course studies. I am hoping that soon we can get to do that. We don't have the time course samples yet, unfortunately.
So one of our audience members is offering you some local San Diego and Baja, Mexico clinical time course specimens, if you would like.
I would love to do that. If you could please send me an email. My email address is, I think, should be at least on the title page of my presentation. Please send me an email. We would love to sequence those.
Great. And I have another question. May sound a little bit out in left field from what you were talking about, but potentially related. So I was struck by the fact that you were talking about 40% of the genome being unannotated and sort of this black box blind spot.
Yeah.
There's a lot of work being done with whole genome sequencing, trying to use it in a clinical context for predicting progression along the continuum of latent tuberculosis to subclinical to active TB. And if 40% of the genome is unannotated, do you think that's a premature usage of whole genome sequencing with MTB? Are we missing something here?
So I think a lot of the conclusions, so let me break that question down. I think that's an excellent question. One issue is that a lot of these studies have been done with short read sequencing. And as a result, certain parts of the genome, especially more prevalently those parts that we don't know much about, are thrown away because of the blind spots issue that I described. So that's one problem. The other problem is even the parts that are not thrown out, because we don't know functional assignment, we don't know what that gene does, we can't actually do a comprehensive conclusion of what is going on. So people report, unfortunately, far too many times conclusions that simply ignore the 40%.
Yeah.
The challenge, and let me describe the challenge and a hope, sort of a light at the end of the tunnel. The challenge is that when you sequence, you're not actually exposing the function, and you do need to do transcriptomics. You do need to do RNA-seq, which has its own very challenging set of issues. Now, the light at the end of the tunnel is that a lot more is known about MTB than is available in public databases. So a paper that coincidentally got accepted for publication yesterday, it has gone, what we did was we went through all the papers and collected all the functions that people had studied and added that to the common knowledge that is out there in public domain. Then we used a protein modeling approach, in silico approach, to see how much of those functions that are already laboratory confirmed we can predict in silico. And we were able to do that quite effectively. And that number, 40%, now needs to change as a result of this publication yesterday. It needs to come down to about 18% because we shed light onto about 20-some% of the genome. Now, the remainder of that, we are able to predict functions that researchers then can go in and specifically test for that functionality. That's much simpler to do than a protein that doesn't have any functional hypothesis, and they have to test for all sorts of functions. So hopefully with this kind of approach, we can get to that end of the tunnel where we have some form of functional assignment for all the genes in the genome. And in addition to that is that I have to say that because of the genomic variability, novel genes are introduced all the time in this species. And as a result, that functional assignment is not something that I see an end to it, but rather an exercise in doing this for every novel gene that we run into every time.
Great. Well, let me see. There's one more question. Have you sequenced and analyzed MTB from clinical presentations of miliary TB versus cavitary? And I think kind of more generalizing that question. So some of your functional sequencing and structural sequencing, do you see a relationship with the type of presentation of TB or the manifestations of TB in those individuals functionally? So
to answer the question is, so let me say that I would like to not answer that question simply because
simply, yes, I could tell that.
Yeah, let me explain why. Because sequencing cavitary isolates is challenging because if you culture it, you are leaving behind a whole lot. And so as a result, the picture is really uncertain. We have developed a sequencing methodology that we can directly sequence from the cavity. A proposal is out there at NIH to allow us to collect cavitary tissue so that we could answer this exact question. And unfortunately, well, I mean, NIH has taken a sweet time to test, to review our proposal. But if, I'd like to make a pitch here, if anyone has cavitary tissues that we could study, we would love to do that and let our new sequencing method that doesn't use amplification also does de novo assembly of the genomes, which would answer exactly this question. We are able to do that. We just need those samples.
Great. Well, we may be able to put you in touch with some of the folks we're starting to work with in the AIDS Clinical Trials Group network, because they're looking at modeling drug entry into cavitary lesions. So we may have some samples that come from cavities.
I would love to collaborate on that, especially related to drug development. We have identified 366 genes that remain identical across all of the isolates, across all of the lineages, not a single mutation in any of them. That's quite remarkable.
That is remarkable.
And that presents 366 drug targets. And if we can then show which ones persist inside the lesion,
yeah,
we have quite a bit to going there.
That would be very spectacular knowledge.
Yeah.
Okay, I think we are right about on time. If there are no other questions from our attendees or panelists, I think we can close, and thank you for what was truly a spectacular talk, very elegant, a lot of hard work.
Thank you. I appreciate it.
Really helpful work. So thank you very much for a great talk. And I'll turn this back over to our hosts, and thank you again.